All right. This meeting is being live streamed. I just got the notification. You are good now. At
Okay.
the end of class, when you end the Zoom, it will automatically upload this video into YouTube, and then you'll do this for fourth and your sixth period. And then what I'll do is tomorrow or later this week, I can help you create a playlist for each class
Okay.
period and put that playlist in YouTube. Upload this video into YouTube, and then you'll do this for fourth and your sixth period. And then what I'll do is tomorrow or later this week. Yeah, go ahead and close that. Otherwise, you'll constantly hear yourself.
Okay. All right. Okay.
Yeah, you'll hear that infinite loop all day today.
Okay.
Mm-hmm.
All
All
righty.
right. Well, that's it. I'm going to run.
All right. Thank you so much.
You're so welcome.
All right. Bye-bye.
Bye-bye.
Okay. Good morning, everybody. All righty. Get everybody on focus mode. Mr. Phillips has fixed me this morning, so we are live streaming today. As of today, there will be live stream videos in case you're absent and miss a class. While we're waiting for others to come in, if you have not uploaded your macromolecule chart, please do that. We are going to do a review today, and then we're going to do a quiz at the end of class. But you'll be able to use that chart if it's completed. So again, while we're waiting, make sure that your chart is complete and it's uploaded. And if it's not complete, be working on it while we're going over the review sheet. Okie dokie. Admitting. All right, almost everybody's here. Good morning if you're just coming in. Okay, let me, let's see. I don't know why it's typing sideways. I don't know why my thing is typing sideways, but it is. All right. Okay. And I think, so while you're doing that, if you just came in, you're making sure that your macromolecule chart is complete and uploaded. And we're going to review first, do a review sheet, and then we're going to do a quiz at the end of class. So you should be good to do the quiz because we're going over all the answers. All right, I'm just sharing my screen. So I'm in Canvas, and if you look at the one that says macromolecule page review, you want to click on that. And then there are two choices of documents you can download. Download the one that says Word document, the one in doc form, not PDF, because you want to be able to write on it. And then when you open it up, it will look like, I'm not going to click on it twice to do it again. It will look like, so again, you want to open the Word one. Oh, it's not showing on your page. Let me see why it's not showing on your page. Nope, it's not showing on the share page. All right, I'm going to stop sharing for a minute and then start again. All right, so again, if you, there we go. All right, so if you download the one that says Word doc, it should look like this. And the top of the page, and we're going to fill this out. We're going to go through and fill this out today as a review for the quiz. The top part is a lot of vocabulary, all the vocabulary that you should know. We're going to, there's a couple of things that we haven't talked about, but I'm going to fill in the answers for you anyway, because let me just find my paper. I got so much going on over here that you're going to need to know them eventually. So, okay. Oh, I know what it is. It's on there. Okay. Got it. All right. Anybody having any trouble downloading that worksheet? So again, you need to make sure that you have, well, you can't see it. Okay. See me. Make sure you've downloaded your worksheet, for those of you who just came in, make sure that your macromolecule chart is uploaded. That's for a grade. Progress reports are going out tomorrow, so you want to make sure all missing grades are done. Not tomorrow, but Friday. Tomorrow's just Thursday. All right, so make sure everything is in by tomorrow. I did put in the quiz that you did last week on quizzes that we did last Wednesday. So that's in. If you missed that quiz, I think I'm going to meet with somebody. I'm available between 2 and 3 this evening and tomorrow evening. So if you need to do that, I think the person who's coming in right now, they need to do that. All right, so I'm going to repeat that. If you missed the quiz that we did last week on quizzes, I can work with you this afternoon at 2 o'clock. What's blocked? Oh, the worksheet is blocked by GoGuardian? Okay. What did I do with the last class? Oh, I uploaded it as assignments. Okay, let me do that. Yeah, it was blocked. So this is what I'm going to do. I'm going to upload it as an assignment. So give me 1 second to do that for you. What is this? This is give me just a few minutes to upload it. And then it'll be in assignments so that you can download it from there. And then that way it'll be in your documents as well. Okay, it has no points. This is not no submission. And it's for today. Today. Okay. Okay. All right, so if you go to assignment, so I'd have to stop sharing. I don't know what's happening today. Today is one of those days. Okay, here we go. All right, so you should be able to go to assignments now. It's published. It's assigned to you. Let me just make sure it's assigned to you. See. All right, so let me know if you can access it now. So you should be able to click on this, download it, and open the document. It's weird that GoGuardian would block it because it's within a program within the. It's taking my computer's slow. Hopefully yours isn't. All right. So is everybody able to see it? Even though mine is taking forever? Because actually, I'm not going to be working on it on it. I'm going to go through a PowerPoint with you with the answers on it. Okay. So let me know if you can't access that, and I'm going to stop trying to open this and then go ahead and access my PowerPoint. How about that? Again, anybody not able, so you go to assignments, it should be there. I mean, actually, it wouldn't be in assignments for you. You don't have assignments. There it is. Finally. Were you able to find it? Was everybody able to, you can shake your head up and down. You got it? Everybody's got it? Good. Okay. Good, good, good. All right. So like I said, I'm going to close that for me. I'm going to open this PowerPoint. Start looking it over. All right, so the first questions, number one, and for most of it, I'm going to go through and just help you with the answer, but I'm going to explain. So you can fill in, but I am going to explain. All right, and some of the things, I haven't had a chance to look at Joe's charts, but the other class that I had, in filling out the charts, some people, the box that has nucleic acids is blacked out for a reason. You should not try to put foods in that box. Notice on this PowerPoint, there's no foods in the listed for nucleic acid, because nucleic acid isn't a food. It's a genetic material found in the nucleus. So you're not going to have anything in that box. So don't try to write over the black box. It's blacked out for a reason. All right, so let's start at the number one. It says the four most common elements for organisms are carbon, and you can write the initials if you know the initials: carbon, hydrogen, oxygen, and nitrogen. Okay, carbon, hydrogen, oxygen, and nitrogen. Okay. And if you don't know the initials and what the initials stand for, then yes, you should write the words out. But CHON is the acronym. CHON is the acronym for organic molecules. CHON, CHON. All right, number two, all organic, and you might want to underline that word organic because that's important. Compounds are made up of one thing. All of them contain the element carbon. Okay, carbon. So if it doesn't have carbon in it, which brings us to the next part, then it is inorganic. So all of the molecules that we're talking about that are macromolecules have carbon in them. Okay, all of them, even the one that's not on the picture right now. All right, for number three, that one you should have listed, and I have it in the order that it's on the screen. So you should have carbon. You need to know that the carbohydrates. Carbohydrates, the monomer for carbohydrates is a saccharide molecule. You can write monosaccharide if you want to, but that is the monomer. A monosaccharide, meaning ones, and actually saccharide means it's a sugar. So carbohydrates are sugars. Okay, they end in, and then if you're looking at the PowerPoint, they end in OSE. And the simplest one is glucose. And then fructose is also simple, and galactose. So those are simple sugars. But the one that we deal with mostly, we're trying to, when we digest our food, we're trying to break it down to a monomer of carbohydrate. We're trying to break it down to glucose. Okay. On your chart, if you haven't turned that in, you want to say polysaccharides for polymers. So if you haven't done that already, that's what should be there, polysaccharides. Examples of polysaccharides are starch, glycogen, and cellulose. And sucrose and maltose are disaccharides, meaning they're just two sugars. But on your chart, you only had monomers and polymers, so that's what you should have under polymers is a polysaccharide. All right, next one, proteins. And the monomer for a protein is an amino acid. And again, on your other chart, you would have had that they are polypeptides, are your polymers, or long chain. The protein itself is a long chain of amino acids. So that's what you should have for polymers on your other chart. All right, then lipids. Lipids, the triglyceride is actually the monomer, and the triglyceride is made up of one glycerol and 3 fatty acids. So you can put that off to the side. So one triglyceride is a glycerol connected to 3 fatty acids, and that's a monomer. This monomer just happens to have 4 parts to it. Okay, so then the polymer would be long chains of triglycerides. All right, so examples of polymers would be waxes and oils. All right, so just remember that butter is, and later on we'll get to that. All right, it says number 4, explain why the structure of a water molecule is polar. I'm gonna come back to this slide in a minute, but I think, all right, this is a water molecule, and I'd like for you to draw it on your paper or somehow add it to your paper. If you can put a diagram there, import a diagram. All right, the reason it's polar, you think of a magnet having a north and south pole to a magnet. So with a water molecule, these two blue parts, this I call it the Mickey Mouse face, the two ears of the Mickey Mouse face are positively charged. So there's a positive end to the molecule, and then notice the red part is your oxygen that is negatively charged. So it has a negative end and a positive end to it. So that makes it a polar molecule. I'm on question number 4. Okay, question number 4 is underneath that chart. Kind of make you want to skip it. But if you can possibly add this picture on there so that and you don't have to put all the little dots in it. What you do need to do is put plus signs over the 2 hydrogens and negative sign underneath the oxygen so that you know that it has a top and a bottom. It has a plus end and a negative end. And so the negative, and just like in a magnet, opposites attract. So the negative end of a water molecule is attracted to the positive end of another water molecule or another substance. Okay, so that's what makes it polar. So again, when you think of polarity, think of North and South Pole. Okay. All right, everybody with me? Am I going too fast? I'm going too fast. Okay, I will slow down. Give you a minute. Oops, I gotta let somebody in. Okay, I think everybody's here now. Might be missing one person. All right. Natalie, good morning. We're you want to try, let me see if I can't back this up just a little bit. Okay, hold on. Nope. Where's the thingy? Oh, you got kicked out. You were there. Okay, that's good. All right, so I don't have to go back. Now, why did it do that? All right, so that's where we are. All right, everybody good? Question 3, what was the fourth macro? Oh, I'm sorry, I did skip that. I did that. I am so sorry. Going too fast for me. All right. Nucleic acid is the fourth one on the chart. That's right. We did carbohydrates, proteins, lipids, and nucleic acid is the fourth macromolecule. And again, on your chart that you're turning in, there's that box is blacked out because it's not a food. It's a genetic building block. Okay. You should have RNA and DNA as your polymer. Okay. So here on the chart, the nucleotide is your monomer. A nucleotide, this word down at the bottom. I'm on the right side picture now where it says nucleotide. So the nucleotide is your monomer. And you can say DNA and RNA are your polymers because they are long chains of nucleotides. Okay. All right. So the box should be carbon, protein, lipids, and nucleic acids. Your monomers are saccharides or monosaccharides, amino acids, a triglyceride, and a nucleotide. Okay. All right. Everybody good? Okay. Now it's the 5 and 6 that we're going to not be super concerned with, but I'll answer it briefly. And that goes back to our polarity question. So number 4 was to know this molecule, molecule of water. There we go. Molecule of water. So make sure that you know the polarity means it has a negative end and a positive end. All right. For number 5, it wants to know a lot of other stuff that we're not going to be concerned about. Universal solvent just means that everything dissolves in water, but we'll talk about that later on. Surface tension, we'll talk about that later on as well. You don't have to worry about that for this quiz. Just know that water is important for life because everything that you digest either dissolves in the water or water molecules are during dehydration synthesis or hydrolysis. Water is either added or removed. So that's why water is important to life. Okay. Everything either dissolves in it. So if you want to write an answer for number 5, it's the last part. Why is it important? Everything dissolves in water and it's important for hydrolysis or dehydration synthesis. You're either adding the water or are people getting kicked out, in and out? Because just added another. Maxwell, you okay? It's like you got kicked out and come back in again. Okay. Alrighty. Yeah, today's a wacky day. All right. Number 6. What you just want to do, you want to do a linear line for number 6. And I don't have and again, this was not something I wanted to spend a whole lot of time on. We'll talk about this one later as well. But a pH scale is just a line, a number line with 7 in the middle. It goes from 0 to 12. So you can create a line like that. It goes from 0 to 12. 7's in the middle. 7 is neutral. So underneath the 7, you want to write neutral. Everything lower than 7 is an acid. Everything higher than 7 is a base. Okay? So that's your scale. 7 is neutral. Anything lower than 7, so between 0 and 6 is an acid. Between 8 and 12 is a base. And again, we'll talk about acids and bases later. There's only one other time, and when I get to that question, I'll bring it up again, but that's your pH scale. Okay. So again, you just want to have a straight line, like a number line, that goes from 0 to 12. 7 is in the middle. 7, and you want to label the 7 neutral. So water would be neutral. Anything less than 7 is an acid. Anything higher than 7 is a base. Things like your orange juice and citric acid, things, those are acids. And soap is a base. Okay. All righty. That brings us to number 7. Everybody with me? All right, so carbohydrates are made up of 3 different elements in the ratio of 1 to 2 to 1. So you have to, the order that you write them has to go 1 to 2 to 1. So carbon, it means that for every 1 carbon, there's 2 hydrogens. That's your next one, and 1 oxygen. So that's what the 1 to 2 to 1 means. For every 1 carbon, there's 2 hydrogens, 1 oxygen. All right. A simple sugar is called a saccharide. And some of these are going to be repeated. Now, the formula for glucose, which is our simple sugar. So there's, you can put glucose on the side as well, or put glucose over the, don't put it on the side, put it over the formula, because that is the formula for glucose, which again is our simple sugar. And it is the product of photosynthesis, which is also broken down for energy during respiration. So when we talked about the equation for photosynthesis, we looked at it forwards and backwards. So you make glucose during photosynthesis, and then that same glucose is used for respiration. We break it down and we give the carbon dioxide right back. Okay. So again, make sure that you make some type of notation that the formula of C6H12O6 is glucose, and it is a simple sugar, and then simple sugars are saccharides. All right, the 4 okay. All right, this is what you want here. And I'll come back to that one later. Number 9, the 4 major polysaccharides in nature. They are starch, glycogen, and you don't have to write all this. You can just write the headings. Now this has 5 because with 4 and 5, if you're a plant, you have peptin. If you're an animal, you have chitin. Okay, so starch, glycogen, which is so when we were talking about carbohydrates, we were talking, and notice these are polysaccharides, so these are carbohydrates. We said that its main function is energy, and we break it down for glucose. And if we don't use all the sugar that we take in, we kind of store it temporarily as glycogen. But glycogen doesn't stay stored. If you don't use the glycogen, it gets stored as fat. Okay, so glycogen is also a storage form of the sugar, but it also says it's quick release. It's not going to be stored for forever. All right, so people who have insulin problems have problems with turning that glycogen back into a sugar, so the sugar stays in their blood. They don't store it. All right, which is not good. Cellulose is in the cell walls of plants. We ingest it for fiber, but we don't digest it very well. So that's why it's good for fiber. If you are, and this is going to come up in another question, if you're an arthropod or fungi, you're going to have chitin in the cell wall because you don't have cellulose, you have chitin for structure. And in plants, we'll have pectin. All right, so that answers number 9. Number 10, okay, number 10 is chitin. Okay. All right. Monomers bound together make polymers. So if you have many monomers, all bound means they're all hooked up together, they make polymers. All right, the three elements that make up lipids are carbon, hydrogen, and you want this to say some oxygen, because if you remember the structure of a fatty acid, it's just long chains of carbon and hydrogen all the way. You have one or two oxygens on the glycerol end of it, but it's mostly carbon and hydrogen. All right, functions. So these are the functions. So you should make note of that. So they supporting cells and aiding essential functions. They help a lot of your structures do what they need to do. The nerve cells have what you call a myelin sheath. So later on when we talk about nerves, we'll talk about the protective covering, like a little sleeve that goes around the nerves is made of fats, helping to absorb certain vitamins. The vitamins that you eat are soluble in the fat molecules, okay? They produce hormones, estrogen and testosterone are lipids. They store energy because of those long chains. What you need to remember too is energy is stored in the bonds between elements. So the fact that the carbon is connected to a hydrogen, that bond is storing energy. In order to get the energy, you got to break that bond. So because fat molecules are large molecules, they are a large energy storage molecule. So after carbohydrates, the main job of a carbohydrate is to give you energy. That's its main job. The second place that your body will look for energy is from your fat. And if you run out of fat molecules for energy, then it will do proteins as a last resort. But that's not the job of the main job of proteins. Main job of proteins is for enzymes and other hormones and tissue structure. But if your body is smart, if it needs energy and you don't have any carbohydrates and you don't have any fats, it's going to go to the protein to get its energy. Okay, structural components. That was the slide I kind of skipped. We talked about, oops, let me go back just a little bit. As far as structural components are concerned with lipids, they are found in the cell membrane. And we talked about the molecule, it's called a phosphobilipid layer, which means, and again, it has two sides to it. So you have the heads, the polar heads. They like water, so they are hydrophilic because they like water. And then you have the tail parts are hydrophobic because they repel the water. Okay. And here again, there's some characteristics of what the lipid bilayer does for the cell. So it's protection. All right, moving right along. Okay. All right, so for enzymes, you need to know one, that they are proteins. That's the most important thing. They are proteins. They are catalysts. So those first two statements you need to know. They're called a biological catalyst because you have catalysts in regular chemistry or inorganic chemistry, but in the body, because the enzyme behaves like other catalysts, it's called a biological catalyst. And what it does is exactly what it says up there, is it speeds up the reaction. So it makes the reaction happen that wouldn't ordinarily happen. So that's what you need for question 14. All right. Question 15, is this as well. It can help build up molecules or break down molecules. That's its job, build up molecules or break down molecules. All right, so an enzyme in your digestive system works on disaccharide maltose. All right, so that means it's working on a sugar, and it breaks it into what is the name of the enzyme. Now, what you need to remember is, and I think it doesn't say it there, all enzymes are named for the substance that it works on, because enzymes are very specific to the macromolecule that it's working on. So there is one for sugar. So they are telling you that this is a sugar, and the name of the sugar is maltose. So the enzyme that you're looking for is maltase. So write that on that line, maltase. All enzymes end in ASE, okay? And they'll be named for the substance that they break down. So the fact that, in this question, they gave you information for a reason. One, they're telling you that it's a disaccharide. So in case you didn't know that maltose was a sugar, they're telling you it's a sugar, okay? So and sugars end in OSE. So somewhere you need to have made a note that sugars end in OSE, enzymes end in ASE. This one is going to be maltase, sorry. Maltase is the enzyme that's going to work on maltose. And the way it does it, and this is not on your question paper, but you need to understand how this works as well, is the enzyme is the thing at the bottom here, the dark structure at the bottom. It has an active site. And notice the active site is shaped like the substrate. The substrate in this question would be the maltose. Okay, so a maltose is a disaccharide. So you've got two pieces to this maltose, and it's going to sit on the enzyme on the active site. And we call this the lock and key theory. One lock, one key will fit. It won't fit anything else. Okay. And once it does that, notice that the molecule changed colors. So it's breaking it up into the two molecules that it's made of, so that by the time you end up with the products, those two molecules are back to being monomers, and they separate and they go their way. The enzyme does not become a part of the product. The enzyme's job now is to go find another disaccharide that it needs to break up. And it goes and it does that, but it does not become part of the digested product. Okay? All right. Any questions about that? Everybody's good? Now, this is where you stop me if you don't understand. You do have a quiz in a little while. So you got to know. You got to know. You might have a quiz. I don't know. So let's see. Yeah, we have time. Okay, we got good time. All right. It says which macromolecule holds your master set of instructions. Underline that part. That's the clue. Master set of instructions are nucleic acids. And they are responsible. They are like the generals of all the cells. They give instructions. They tell it what to do, how to do it, and when to do it. Okay. Number 18, factors which limit enzyme activities. This is where our pH comes in. So you want to put pH here and temperature. pH, most things, your blood needs to be a certain pH. It's kind of on the neutral side, slightly acidic, but neutral. Enzymes are proteins. They don't like being in acids, and they don't work well in bases. So when they asked it what limits their activity, they need a proper pH, and they also need a proper temperature. Remember, I talked about the fact that if you get a fever, your enzymes don't work. Okay, and that brings us to the next question, denaturing. These little molecules down at the bottom are shaped to fit what they work with. Denaturing changes the shape of the enzyme. Okay, high temperatures or too much acid will change the shape of the enzyme or the protein, any protein. Other proteins in your body don't function well in poor pH or high temperatures. So if it's not shaped properly, then it's the key or the lock is not shaped, then the thing that's supposed to fit it won't fit. Okay, it won't work. All right, I'm going to give you a few minutes to look at the true and false section, and then I'll come back and talk some more, but just a few minutes because we're going to do the quiz in a little while. And you can't start the quiz. I have to give you a code for that, so don't jump ahead and try to start the quiz. But take a few minutes. Oh, actually, this is question 20. Question 20. Again, I talked about the fact that all these molecules have a bond between the carbon, the hydrogen, and the oxygen. In order for us to get energy, we've got to break those bonds. And so the two molecules that you want are ATP and NADPH. They are energy carrying molecules. And later when we talk about respiration, we'll talk a lot about them, but it's ATP and NADPH. Those are capital letters, ATP, capital letters, and NADPH, which will help you with doing the true-false questions, because they ask you about both of those. All right, so I'll give you the next few minutes to do that, and then we're going to do the quiz. Okay, I'm going to stop sharing. All right, true, false. So I'm going to just go down the line. I'm going to give some explanation, but I'm going to go down the line. So the first one says active site would be found in a lipid. Active sites, as we just saw in PowerPoint, are found on enzymes, and those are proteins, so that's wrong. That's false. Enzymes are very specific in how to their substrates. That's true. That's our lock and key theory. So if you want to make a note of that, lock and key, that's what we mean by very specific. All right, unsaturated fats are liquid at room temperature, and that's true. There are two types. There's saturated fats, and those are like butter, which are hard, butter and lard, and unsaturated fats, which are liquid at room temperature, like oils. Nucleic acids are the main source of energy. No, they are not. Those are your genetic materials. Denaturization allows the enzymes to be recycled. They don't, because the enzyme, a denatured protein or a denatured enzyme is no good because it's lost its shape. So it cannot be reused because it's out of shape. So that's false. Phospholipids are an important component of all cell membranes. That's true. That's that picture I showed you. Amino acids is to protein as a simple sugar is to starch. That is true because amino acids are the building blocks of protein and sugar is the building blocks of starch because starch are large molecules. All right, and here NADPH is a lipid. That's false. I just told you it's the energy carrying molecule. A substance with a pH of 3 is a base. That's false. It's an acid. That's why you drew your timeline. All right, animals store glucose in the form of glycogen. That is true. All right, going on the next one, and I'm just going to do the next section, and then we're just going to go do the quiz. All right, denaturization would be P for proteins. ATP would be C for carbohydrates, and I'm going to leave it at carbohydrates because it's not really carbohydrates because it's an energy molecule. So I'm not really wanting to give that a label, but if we're going to give it a label, we put it with carbohydrates because it's the energy carrying molecule. All right, triglycerides is lipids. Primary source of energy is carbohydrates. Sugars are carbohydrates. Polypeptides are proteins. Fatty acids are lipids. Um, Nitrogen base are nucleic acids, and peptide bond holds amino acids together, so that's proteins, and enzymes are proteins. Okay. All right, so I'm sharing my screen. So if you go back to Canvas and you look at your module, the last thing on your module says Biology Unit 1. There's one. Yes, I know I'm going to skip that one, so don't worry about that. Don't worry about that section. And then this is a review page, but I want you to get ahead and have enough time to work on this quiz. There's only 15 questions. It shouldn't take you long, but it is giving you 30 minutes to do it. It is timed. All right. So you should, I look at it, student version. It should look like that. In order to take the quiz, and I'm going to type it needs a code. So I'm going to type the code, and it is case sensitive. So you need to type it just like I type it. Oh, it's not. Oh, I'm typing it over here. Okay. 1, 2, 3. Okay. So I just put the code in the chat, and I guess I can type it up here. Oh, see. I go ahead and start as soon as you get it to open. It tells you that you have 30 minutes to do it. You're welcome to log out when you're done, but only log out if you've already submitted your macromolecule chart. If you haven't finished your macromolecule chart, that's due today. You can use your study guide to help you. Attendance. While you do that, I need to do attendance. Mhm. I thought Landon was here. Hmm. Again, when you're done, you are free to go. Have a great weekend. Do I see you again? I see you again. No, I see you Friday. I don't know why this feels like Thursday. Friday, Friday. See you Friday. And again, don't leave if you haven't, and a lot of you still haven't uploaded your chart. So make sure you upload your chart, please. Fine.
Ma'am, did we turn in our study guide?
No, that's for you. So save that. Don't lose it.
Okay. And I have another question. Are we able to retake
Were you able to do what now? You're breaking up on me.
the test?
This one for today?
Get another?
Did it not allow you
Yes.
2 times? No, okay.
No.
All right, let me, I'd have to give you another code for that, but yeah, we'll have a day when you can do that. So do you want to meet this afternoon for the other, or you want to wait till tomorrow?
What was the quiz called? Like the one that we were
It
supposed to take Friday?
it was just on protein structure, so it was just about proteins, and so some of the things that we talked about today.
Yes, okay. I would prefer if we met today so I can get it over with.
Okay, so it's just on proteins. All right, and it's a quizzes, so I have to give you when you so log back in to the Zoom, and I'll give you a code, and you can do that. So say, let me check. I think you need to do this as well. So it'd be nice if both of you did it together. Are you the one? Yeah. So can you do it today at 2 o'clock? Say? The quiz that you were absent last week when they did a quiz on quizzes. So I have to give you a code for that. So if you can log in today at 2 o'clock, can you do that? Then I can do both of you at the same time. Okay. So I'll be here too, and just log back into the Zoom, and I'll get you set up with that. Okay. Okay. She may not like the score of it.